So welcome everybody. My name is Josh. I'm a cryoEM scientist from the University of Leeds. There's a good number of you in the chat now, so I want to welcome you all to the 20th webinar of what has been a really interesting series called Structure Meets Function, put together by Instruct. So there might be some of you joining today who aren't very familiar with Instruct-ERIC, so I wanted to start with a couple of slides introducing Instruct and what it's about. So Instruct is a pan-European organization which facilitates access to structural biology infrastructure. There are 14 member countries, each of which pay an annual subscription to allow scientists working in those countries to access the full range of services and opportunities that are available through Instruct. So the core offering, or one of the core offerings, is research visits. And these are a fantastic opportunity for researchers to access a whole catalogue of equipment, expertise and resources to facilitate structural molecular biology research. So this is anything from access to sample preparation facilities all the way through to time with equipment such as cryo-electron microscopes. And there's a huge variety of different techniques. So you can see here over 75 different services. And the aim of this is to encourage a multidisciplinary approach to structural biology research. Beyond facilitating access to technologies and services, Instruct actually offers a lot more. So one of the big things is training courses. So they offer training in a range of core and emerging methods, including hands-on workshops delivered by international experts. So these courses can be a great introduction to new techniques. So do keep an eye out for those. In addition to the training catalogue, Instruct offers an internship program. So this is great for cases where researchers are interested in gaining deep insight into particular techniques, or maybe a project requires access to equipment and expertise over an extended period of time. And the internship program funds research visits for 3 to 6 months that allow a person to really become embedded at that site and gain a more in-depth training and experience in the technique or techniques that they're interested in. And then finally, Instruct offer direct funding for small-scale pilot research projects in integrated structural biology. And this is great because these awards can allow researchers to get the preliminary data that they need for grant applications to gain further funding. So a whole range of different activities and opportunities offered by Instruct that you can benefit from. And I highly recommend that you go and check out the website to read some more about those. As I mentioned at the beginning, I work at the University of Leeds, specifically within the Asbury Biostructure Laboratory. So I want to tell you a bit about what's going on at Leeds. Among the range of facilities within the university, we're really excited to be able to offer 3 of these technologies, so mass spectrometry, NMR and cryo-electron microscopy through Instruct in terms of inviting users onto our site and working with you on these projects. So to briefly introduce these capabilities, cryoEM access at Leeds, we have access to our 2 Titan Cryos microscopes, which now have shiny new Falcon 4i detectors. And we're able to cater for experienced users who may just want to send a sample, and we can then facilitate high resolution data collection. But probably more excitingly, we're able to provide enhanced support in going from sample to structure through Instruct. So for projects where maybe you just have some biochemical analysis of your sample and you've never done electron microscopy before, we can take you through the pipeline from negative stain, cryo-EM grid preparation and workup all the way through to high resolution data collection. So this is a great opportunity for researchers who aren't experts to gain cryo-EM access to cryo-EM workflows. And then very quickly, I wanted to highlight the mass spec and NMR facilities. So within mass spec, there's a range of technologies available, but in particular, we offer hydrogen deuterium exchange analysis through the Instruct catalog at the moment. And then in terms of NMR, we're quite lucky to have a 950 megahertz magnet and to offer some interesting and fairly unique capabilities, all of which are again available through the Instruct catalog. So if anyone's interested in cryo-EM or mass spec or NMR and wants to talk more about their project, then we'd really love to hear from you. My email address is at the top, our cryo-EM email address is in the middle, and our general facilities email address is at the bottom there. And then just as a last note, I've been asked to flag another great opportunity. So the first ISADOR call for projects focusing on SARS-CoV-2 research is now open. So ISADOR, which stands for Integrated Services for Infectious Disease Outbreak Research, is an EC-funded project that will provide access free of charge to research infrastructure services, such as Instruct structural biology services. And this is specifically for transnational access, so for services outside of your country of residence. And what's great is that scientists from both European and non-European countries are eligible. And this includes scientists in both academia and industry. So regardless of where you are or what you're doing, the call is open to you. And if you're interested, all the information you need is on the slide. And that's it for the introduction from me. We've got two great speakers today who have both made use of the cryoEM facilities at Leeds. Just a note, we'll be taking questions from the Q&A section at the end of each talk. So please try and put any questions in the Q&A rather than the chat. And yeah, without further ado, it's my pleasure to announce or introduce our first speaker, Jamie Blaza from the University of York, who's going to talk to us about a bifurcating hydrogenase from Thermotoga maritima. So Jamie, if you want to go ahead and share.
Brilliant, thanks Josh. Brilliant, yeah. So hello, I'm Jamie Blaza. I'm based in the York Structural Biology Labs in the Department of Chemistry at the University of York. And I'm going to be talking a bit today about understanding the mechanism of the bifurcating hydrogenase from Thermotoga maritima through the application of cryoEM and symmetry expansion. And I will dwell a little bit on symmetry expansion because I think it's a really powerful tool. It's one of those tools that is used lots in the virology community. But actually, even if you don't have such high symmetry as a virus, symmetry expansion can be really, really useful for you. And I think it's something that maybe a few projects are missing that they could apply and get further information from their data. So before we get into the content, it's just to say that this work was led by Chris Furlan at York and Nipa Chongar at MPI. So Nipa did the biochemistry, which for this type of enzyme is non-trivial, and Chris did the cryoEM and data processing. And also just to say that our paper is currently up as a preprint on Chem Archives, although I hope it's published soon. It's been a bit of a tortuous route for us. So the biological context here is Thermotoga maritima carbohydrate metabolism. So Thermotoga is a hyperthermophilic anaerobic bacterium, and it really likes to oxidize sugars to acetate. And during metabolism of glucose, for instance, both NADH and reduced ferredoxin are produced. Ferredoxin will very readily pass electrons to protons to make hydrogen. It will thermodynamically, it's ready to do that. But in some conditions, the reaction of NADH to donate electrons to protons to make hydrogen is not as favorable. And so the solution is to combine the two reactions in what's called a conficating mechanism. And so we'll dwell on this a little bit here. So here you can see the redox energy of the different couples, and you can see that the proton to hydrogen couple is in the middle between ferredoxin and the NADH couple. And essentially what can happen is that you can either run a confication mechanism where ferredoxin passes electrons downhill, and the energy released from passing electrons downhill is used to boost electrons from NADH uphill, to reduce protons to hydrogen. So you're sacrificing one reaction to drive another reaction. Or you can do the reverse, where hydrogen donates 2 electrons to the system, and one goes up to ferredoxin and one goes to NAD+. And if you do that cycle twice, you'll fully reduce ferredoxin and NADH. So it's a really clever little bit of redox mechanism here that's really quite fascinating. The sort of relevance of the enzyme is that there's a growing body of work looking at the synthetic biology of electron transfer pathways, and historically it's not been so easy to engineer electron transfer pathways, often because you've got complicating cofactors which you need to incorporate and you need to make assembly pathways, but that's coming on really nicely. And the nice idea here is that reduced ferredoxin is a very powerful reductant. So it's a little bit like biological borohydride being a common chemical reductant that's used in synthetic chemistry. And so using these common biochemical intermediates, NADH, which is in all organisms, and hydrogen, which because of the growing hydrogen economy, probably not produced biologically, but produced non-biologically, you can actually on demand make reduced ferredoxin to power different reactions. And actually, because reduced ferredoxin is so ready to donate electrons, it can convert carbon dioxide into reduced forms of carbon dioxide like methanol. So before we get on to our data, I just wanted to dwell momentarily on a much better studied bifurcating enzyme. So in this case, it's the BC1 complex. And what happens in the BC1 complex is you're taking quinol, which I've indicated here as QH2, and you take one electron and it goes down a pathway to this ISP, which is the iron sulfur protein, onto another a C-type cytochrome. And that happens thermodynamically downhill. And the quinol becomes a semiquinone. And then the ISP, which I've indicated here as a diamond, moves away, it diffuses away. And because it's diffused away, it's no longer within electron transfer distance of the semiquinone. And so the second electron, which the semiquinone would like to donate readily, cannot go down the downhill pathway. So instead, it's forced to this other quinone site on the other side of the enzyme, where a quinone is reduced to a semi-quinol. And then after a while, the ISP diffuses back and the cycle repeats itself. But what's really important here is this movement of the iron sulfur protein, which forms a gate, because thermodynamically, everything just wants to flow downhill. But because you have this gate moving out of the way, and then after it's done the second reaction moving back, you're gating the electron transfer. So one goes downhill and one goes uphill. And the nature of this conformational change was actually originally shown in the late 90s with some really elegant anomalous scattering experiments looking at the movement of the iron sulfur cluster. So that's a sort of model system that we can all bear in mind as we start to think about how this enzyme might work and how the structure will hopefully illuminate function. So the key questions here, the protomer is 150 kilodaltons, but that we know has to become a 600 kilodalton tetramer from other experiments. We need to know how the redox active sites are arranged and how they allow catalysis. And we need to know how is bifurcation enforced. So before we started with the structural studies, the group that we were collaborating very closely with, led by James Birrell and Hideaki Ogata, doing biochemistry led by Nipa Chongar, who I introduced in the first slide, had already developed a really nice heterologous expression system for the bifurcating hydrogenase, where they make it in E. coli, and because E. coli doesn't have the iron-iron active site incorporation system, they then add that after purification. And they then characterize their enzyme really with a tour de force application of bioinorganic techniques. These are very specialized but extremely powerful techniques. And they did a little bit of everything, it felt like, apart from structural studies, which is absolutely brilliant to have collaborators come to you with such a well-characterized, fully active, fully pure source of enzyme to do structural studies. We started, one of the active sites that we know has to be there is an iron-iron active site, and that's extremely sensitive to oxygen. And we originally tried to prepare grids either with the iron-iron site in to make the apoenzyme, or the iron-iron site out to make the apoenzyme, or the iron-iron site in to make the holoenzyme. And whenever we did the holoenzyme, the grids didn't work. And we did this grid screening in Sheffield. And so we decided, let's focus on the apoenzyme lacking this oxygen sensitive site, because we didn't have the techniques to make anaerobic grids. But we got these screening results in Sheffield, and you can see a progress report from James in the bottom right there from late 2019, where we realized it had to look like a frog from these 2D class averages. So this was the point at which we applied to Instruct using these 2D classes as good preliminary data that we had something worth collecting on, and we got access to the data collection to do data collection on the cryoses in Leeds. And I had a very weird drive over to Leeds on the 18th of March when the whole world was just going into lockdown. Thankfully, when we didn't have any time to pre-screen these grids, we just thought, okay, well, things are going weird. Let's just make what we have and go. But thankfully, with a lot of help from Becky and Charlie, who I'll acknowledge at the end, we were able to image we got an absolutely beautiful data set and immediately could see these fantastic 2D class averages with really high resolution features. And we were instantly confident that this project was a goer. So this is how the enzyme looks. You can see that the frog has become something much more defined. It's a tetramer structure around this core, and the core is made of the HID-A enzymes. The nice thing about cryo-EM is the iron sulfur clusters stick out like a sore thumb. So we could immediately see the iron sulfur clusters, and these iron sulfur clusters are what make up the redox electron transfer network, because they're all within about 15, 16 angstroms of each other, which allows electron transfer. We could also see the other active site that makes up the flavin. That showed up very nicely in the density. It's a nice big organic molecule. And so we also realized that the way the enzyme was multimerizing is you had a core of HID-A proteins and then elaborated off the HID-A tetrameric core, you had 4 HID-B, HID-C domains. And really each HID-C and HID-B domain just is interacting with one HID-A core element. And we also realized that there were 2 electron transfer networks. You can see at the bottom left there, you can the distance between the 2 networks, which are made of iron sulfur clusters very closely together, is too great to allow electron transfer. So there must be sort of 2 independent halves of the enzyme that probably just operate completely independently of each other. We also realized that within each independent redox network, there was a connection via a bus bar that connected one HID-B, HID-C side to the other HID-B, HID side through a HID-A core. And actually, this was really nice to see because I already introduced the BC1 enzyme, but the BC1 enzyme has a very similar so-called bus bar topology of its redox networks. So the resolution revolution continues. I'm very fond of this image because it just shows you how beautiful the data is, because we collected it on very powerful microscopes with really nice detectors it leads. And so here's a little bit more figures, and more people, I would hope, would do this, where you don't just show density from the very best bits of the map, but here we've shown density from some of the best bits from the map, such as the iron sulfur cluster A3, which was shown in the top left, but also in the weaker bits of the map, such as the iron sulfur cluster B4, which is in panel D. We could also see some waters, which was brilliant, and we could also see, if you squint, sort of some holes in the phenylalanine aromatic residues. And this is on the K2 detector, so we're obviously very keen to prepare some more protein, send it back to Leeds to see what we can do with the really nice new Falcon 4i detectors. But the resolution of the core was about 2.3 angstroms, which we were really pleased with. So the first thing we noticed was a very clear relationship between these enzymes and an enzyme called complex 1. So complex 1 is extremely well characterized redox enzyme. It doesn't bifurcate, it just has an NADH module that is oxidized by the FMN and then passes electrons down this iron sulfur cluster chain to pass electrons to quinone to power mitochondrial energy metabolism. And we could see actually that complex 1 has still got the iron sulfur cluster that makes up the iron-iron active site in our system. But it's missed one of the connecting iron sulfur clusters, which was really interesting to see. And we could also see that where complex 1 passes electrons down to this quinone substrate here, in the bifurcating hydrogenase, you can see that there's a connection made to the other protemer, this so-called bus bar. So you can see how complex 1's a monomer, so you can immediately see how multimerization of this enzyme is actually changing the electron transfer pathways. The other implication of this multimerization of the enzyme is this CPI system, which is a well-characterized hydrogenase. It doesn't bifurcate, it just takes electrons from reduced ferredoxin, passes them to protons to make hydrogen. But the ferredoxin site that we see in CPI is actually occluded, again, via multimerization in the bifurcating hydrogenase tetramer. So, and doing all this processing in the first lockdown, lots of time to think and bang our heads on our desks in our, respective rooms where we were sheltering. We see these electronically connected active sites, but there's no hint of how they make any kind of bifurcating system. And you can also see that the core of the enzyme, made of HID-A, is really beautifully resolved, but the outside lobes are very much worse. And so because the outside lobes are much worse, we immediately thought, oh, there must be some unresolved heterogeneity here. It could be, an interesting flexible conformational change, or it could just be, the enzyme falling to bits. But we could also see the first inklings of some density between the two lobes. And you can see that at the top and the bottom of the local resolution figure there. So, yes, we settled on symmetry expansion. And the reason we settled on symmetry expansion is because we had the HID-B and HID-C lobes coming out from the HID-A core, we knew that classification would be really tricky. If I just move this screen, yeah, so if we just classified, maybe focusing on where we think there's heterogeneity there, because we have a tetrameric complex with a flexible domain, but each flexible domain is probably independent of all the other flexible domains. You would have to rely on, or whatever other software you're using, with 81 different states, assuming that each flexible domain in the tetramer is in one of two states. And so that is not much help. Not only do would it be computationally inaccessible, of course, if you've got a reasonably sized data set, by the time you split it into 81, suddenly you don't have very many particles left. And so that's a problem. So how do you get around that? Because you have symmetry, and the problem here is that you have symmetry being enforced. So instead, rather than just classifying the whole particle, we transform the particle with a symmetry operator. Here, this is assuming a tetramer, so this would use a D2 symmetry operator. And you create a mask that just covers a single bit of the tetramer. So in this case, because we have tetrameric symmetry, we would apply the D2 symmetry operator, and then we would then end up with the number of particles times 4 subparticles, each transformed by this operator. But you can then focus in on the individual domain that you're interested in within, and because you have transformed your particles by the symmetry operator, the individual domain, the individual domain has a subunit from each of the 4 tetrameric areas in it. So you apply the mask, and you now only have to have, assuming there's 2 different states, you only have to have 2 classes, and you don't need to split your data so much. So here you've expanded the data by the symmetry, and you've then focused in on a particular area using a mask. And this is a technique, as I mentioned in the introduction, used an awful lot in virology, but it can be applied to any kind of enzyme or any kind of target that has symmetry. So this is our classification regime, and we started with particles extracted from a map that was refined with D2 symmetry. And originally I actually tried to use D4 symmetry, and that didn't work. So instead I moved to C2, and I'll mention why that is when I show how our results and why we had to do that. So we start by creating a mask, and because we could see this fuzzy, messy density between the 2 lobes, our mask just focused on that area. And we then did C2 symmetry expansion, and then 3D classification with a mask just over that particular area. And then we could immediately see these bridge domains forming between the lobes. And because there's there's two lobes, but each lobe can individually form a bridge, we had two classes, one with the bridge at the front of the complex and one with the bridge at the back of the complex. And we then created a mask of the whole C2 domain and refined that to a resolution of about 2.8 angstroms. We could still see a little bit of fuzzy density in the particles that didn't have this bridge forming. So we could actually focus in a little bit using a mask on that area as well. And you can see that going down the right of the figure, where and again we used a focused mask to find the particles within a particular conformation, and we used a larger mask to refine them to reasonable resolution. The reason that D2 symmetry didn't work was that this bridge forming breaks the D2 symmetry, and therefore really we're using C2 symmetry, which actually is quite elegant, because if you think about how this enzyme is broken into two redox networks, the C2 symmetry applies to each of the redox networks. So essentially, if we have two independent enzymes within a single complex, using symmetry expansion, we're really just focused on a single enzyme. This bridge is formed from the C termini of hid B and hid A. And the reason we're very excited about it is that this bifurcation reaction is known to only occur with enzymes that have this hid B extension. The hid A extension is also only found in enzymes that bifurcate, but there are some bifurcating enzymes without the hid A extension. But all bifurcating enzymes have this hid B extension. So immediately this was really exciting, because as I introduced with bifurcating enzymes, you need a gate. You need something that stops electrons just flooding thermodynamically downhill. And this kind of conformational change immediately is a candidate for such a gate. I introduced how we could also look at the class of enzymes that don't have the bridge forming, and we found a low resolution position for the hid B extension. It didn't refine as nicely. It's probably very flexible. But looking at these two states, we immediately have a conformational change that would be consistent with some sort of gate, with either this hid B domain up or the hid B domain down forming a bridge. And there's a movie here that illustrates this change. So there you have it in the two states, and you can also see in where their domain joins the rest of the lobe, the zinc that we have found, which we think is acting as a sort of pivot. So I'll just play that you again, because I think it's really the key finding of all this work. So there's the conformational change, and we're now going to look at the conformational change from a different angle. So we then started to think about electron transfer networks, and again, using this principle that redox factors have to be within a certain distance of each other to efficiently pass electrons. And we could see that the only, we have what we think is a zinc, the coordination of that mystery metal site certainly matches a zinc. And when we look at just the hid B enzyme, we do find the zinc, a zinc molecule in there, in addition to a lot of iron because of the iron sulfur clusters. And so we could immediately start to try and push the data into some sort of mechanistic hypothesis. And, you know, I'm going to be completely honest, we haven't completely solved the mechanism quite yet, but it's given us some leads. Currently, our idea is that if we're looking at electron bifurcation, you'll first have the hydrogen reacting at the H cluster. We know where the H cluster is because very, very similar enzymes have been solved. So even though we didn't incorporate the H cluster, the iron-iron site I was talking about earlier, we can infer where it is. And that will donate various electrons to the redox network. And then some of those electrons will be passed up to the bridge domain, which we think must be in some third position that we haven't yet detected to be able to accept electrons. And the bridge domain then moves, ferredoxin can come in and take those electrons, and then the enzyme is reset, ready for another go around. And as we were well, we put this paper on, just as we put this paper on Chem Archive, really, this other interesting paper came out, which looked at a related enzyme that is, rather than an iron-iron hydrogenase, it's a nickel-iron hydrogenase. But the actual core enzymes are very, very similar. Rather than making a tetramer, this just makes a dimer, and there's no sort of bus bar element. But in all the processing, C2 symmetry was enforced, and this meant that the conformational change wasn't quite as readily disentangled, but it was certainly, they saw a very similar domain movement in the HID-B domain, which I said was exclusively found in bifurcating systems. And so it'd be really interesting for the authors to revisit their data set using symmetry expansion, so that they can maybe disentangle their own conformational change. So I just have a few closing thoughts. We've now got a growing family of structurally characterized redox enzymes, and it's clear in this family that multimerization on evolutionary times completely changes the networks. It either blocks or it enables active sites. It allows very different mechanisms. We've found a strong candidate for some sort of gate conformational change in this bridge domain. And the scene is set for serious falsification of working hypotheses of how we think the enzyme is functioning. But what I would really hope this audience takes home is how symmetry expansion can be really useful outside studying virus capsids. It's a really powerful technique, and it doesn't actually matter whether you enforce or don't enforce symmetry in your refinements. Either way, symmetry expansion is a way to dig deeper into your data and try and understand how the protomers behave. So just a few moments for the final acknowledgement slides. So on the left, you see the whole team, Chris and Nipa, Pooja, Wolfgang and Hideaki helped along the way. And I work really closely with James Birrell, which is very nice because we did our PhDs together. So to now be collaborating as independent group leaders is a really nice circular feeling. I'm enormously grateful to Becky and Charlie for getting our grids on the Cryos just before the first lockdown. You can imagine, as a young group just trying to get going, us being able to process our first data sets in the lockdown rather than just twiddling our thumbs really made a huge difference to morale. And so, not only was the data set fantastic, but Becky and Charlie made a huge difference, well, just to our lives in those weird months. And Svet helped us screen our first set of grids in Sheffield, and computing resources was provided by Viking HPC. And finally, I should thank Instruct for providing us with the microscope time and supporting me by inviting me to this webinar. Thank you.
Thanks, Jamie. That was a really, really interesting talk. And while we're waiting for some questions to come in, I think I'll take moderator's privilege and ask a couple. So the first one is a little bit more general. So you mentioned being excited about the new detectors at Leeds and potential improvements in resolution. I was just wondering, what information might you be missing, or are you looking to get by kind of modest improvements in resolution, or is it just tidying up interactions with iron sulfur clusters and getting distances more precise and things like that?
And so actually here, the resolution you need has to be sufficient to answer your interesting and hopefully relevant biological questions. So for redox enzymes, distance and exact conformation is really important. And so for us to sort of start looking at the iron-iron active site at high resolution, of course, quite a lot of work has been done looking at some of the new data sets at the kind of well, of course, the famous one is the apoferritin at 1.2, but others are starting to come in at more of in the sort of 1.5 to 1.8 range using these new, very powerful detectors. And particularly in those systems, you can refine you don't refine the hydrogens, you refine the data against your model, but you can see the hydrogens appear as anomalous density. And so for something like hydrogenase, seeing what's happening with your hydrogens is incredibly important. And because it is a thermophilic enzyme, and because, at the moment, we have literally only seriously collected one data set, hopefully with a few more optimizations in the prep, combined with the improved hardware at Leeds, I'd hope we could get this down to like 1.8, 1.7. And yet, particularly if we use our anaerobic Vitrobot, which we're setting up in York, and is maybe something that might be useful for a few people, we can get the ion site in there and really create a complete chemical picture of what's happening there. So yeah, we do have specific scientific questions that would be answered with improved resolution.
Yeah, that makes perfect sense. And given that I can't see any questions yet, I'm going to take second moderator privilege and just ask a more specific question. So in your HID-B extension that had a slightly lower resolution, I think it was the HID-B extension, I was just wondering if you tried a focus classification without alignment approach on just that region.
Yeah, so actually a lot of our classifications were done without alignment because that core is so solid and so well behaved. You can get your original angles really accurately just on the core, and then you don't need to do more do alignments in your classification. We did get slightly better resolution when we did them in refinement, I think, although I have to check with Chris, but certainly it behaved very nicely. The key with that HID-B domain being up and being in poorer resolution actually is because iron sulfur clusters, particularly 4 iron 4 sulfur clusters, scatter so strongly, they show up like a sore thumb in your maps. So we can be pretty confident. I mean, we didn't build it with side chains. We just built the backbone and the iron sulfur clusters, but we're pretty confident that it's in a reasonable position.
Yep. That makes sense. I mean, if that's all the questions and no one's got any burning questions coming through, obviously you can leave them in the Q&A section for Jamie to answer later. But I think we're gonna move on to our next speaker, which is Professor Colin Berry from Cardiff University, with a talk on insecticidal proteins, specifically VIP3, I think. So if you want to share and take it away, Colin.
Great, thank you. Excellent. Okay. Well, so yes, as you've just been told, I'm going to talk to you about insecticidal proteins. I'm not going to talk to you about this from the point of view of the techniques of structural biology, but the lessons that it has taught us in terms of the way that this particular protein is likely to function from basically a zero start. And I apologize because I've just seen a horrible typo in my title on this slide. So moving on, I work in the field of insecticidal toxins. These are protein toxins produced by bacteria that will kill specific but small groups of insects. And as you can see here, there's lots of different structural types of protein which are known, which will kill insects. And 1 or 2 of them are enzymatic toxins, but most of them are pore-forming toxins. And I'll just draw your attention to this guy in the center as a particular example. It's the best known class, the cryotoxins. And it's a toxin made up of 3 different structural domains. And I'll be coming back to cryotoxins later on. We've actually been doing some work in Cardiff and elsewhere to classify insecticidal toxins, and they're all listed on this webpage with a new classification system. Okay, so these toxins give us the ability to control insects in a very environmentally friendly way. And I hope I don't have to persuade you of the importance of insects as pests, particularly in agriculture, but also in human health, where they often vector diseases. And clearly we can control insects with chemical pesticides, but problems are ever present with this class of chemical agent, because we have a lot of insect resistance that arises in the field, and there's a particular issue with toxicity to non-targets, friendly, beneficial insects, and so on. So what we want is an agent which is far more specific than these chemical pesticides, which does not have non-target effects. And that's what brings us to these biological agents, because they have specificity, and the producer organisms, which can be used as sprays in the field to control agricultural or human health pests, often act by multiple mechanisms. And in addition, very commonly, we can take the genes that encode these proteins and put them in transgenic plants so that the plants are thereby insect resistant. So as I've said, many of the toxins that we work on are pore formers. And just to give you an insight into pore forming toxins, they usually fall into one of two groups. The alpha pore forming toxins, and this particular protein APP6 is an example of that. And what happens is during the mechanism of action, we think that this region unfolds and inserts in the membrane with a lot of its friends to form a big multimer oligomeric pore like this, with just the tips of these loops inserted into the membrane, which I characterize a little bit like this shuttlecock, with most of the structure standing proud of the membrane out here. So there's our alpha pore forming toxins. There's also the beta pore formers. This is lysenin, which comes from the earthworm. I don't have an example for a bacterial insecticidal protein, but these unfold to some degree in this body region down here to form this long stem, which forms an interior pore when it inserts through the membrane. So those are the two general classes. The protein I want to talk to you about, or the family of proteins, is the VIP3 family. And this was first discovered in bacteria in the mid-90s. And it's produced during the vegetative phase of growth of a very common insecticidal bacterium, Bacillus thuringiensis. And it had no homology to any known toxins, so no clues there about how it might work. They're active against a very wide range of Lepidoptera. Those are butterflies and moths, so caterpillar pests of agriculture. The protein itself is approximately 90 kilodaltons, but it associates naturally when you produce it into a tetrameric form. In the insect gut, it gets processed to approximately 65 and 21 kilodaltons, but the two fragments remain associated. So it doesn't lose part of the protein at that point. It just remains attached, but proteolytically activated. And we can mimic this processing which occurs in the gut with trypsin in the lab in a slightly more controlled fashion. It's clear from pore formation assays that that is its mechanism of action, that it can form pores in membranes. And as I mentioned before, the beauty of some of these proteins is we can put them in transgenic plants to make them insect resistant. And it's been quite widely deployed into things like corn in order to convey some insect resistance. So a very exciting protein, a new generation of protein for use in the field, but absolutely no clue about its mechanism of action. So on the structural front, clearly we want to understand what it looks like so that we can predict function. And the initial good news is that it crystallizes easily and you can get these beautiful looking crystals. The bad news is that their diffraction is awful. Try as we might over many years, we've never been able to get anything that diffracts better than 8 angstroms. And so crystallography is not the way forward. And therefore we came to electron microscopy, and we did some initial work just with some TEM, and we managed to get some interesting preliminary work that showed us this surface topology with a clearly tetrameric form with these wings coming away from a central core. And this also gave us hope that we had good enough quality material that we could get into some cryoEM and actually solve the structure in a serious way. And with collaboration with Leeds and with partial funding along some parts of this process from Instruct, we've managed to get some structural data, which I will share with you. So having started with no structures, there was then very recently a whole raft of structures came out. One of which was ours, which is the one I will talk to you about in most detail, this VIP3BC1. We were slightly pipped at the post by another cryoEM structure for the VIP3A variant. And there was also a mutant structure produced of the same protein we were working with. And by making a high number of mutants, they managed to get it to crystallize and cooperate, and they've got this structure. So you'll see that they're all very similar. I will talk to you in detail about this one. All of them have this tetrameric form, of course, and you can see still, as in the TEM, the wings and a central core. And for those of us who are old enough to remember old arcade games like the Space Invaders, back in the day, it's been characterized as looking like a Gallagher spaceship. And so it's got this very interesting structure with the central peak. And in fact, although it's tetrameric, it's clearly made up of a dimer of dimers in the structure. And that sort of interaction has been lost somewhat in the mutant, which cooperated on the crystallization. So if we just compare quickly between the VIP3A and the VIP3B, what you'll see is that most of the structure with the VIP3B in color overlays very well, except this domain down here, where in the VIP3A there's it's more linear with the rest of the molecule, and in the 3B it's offset somewhat. And the reason for that seems to be that in the VIP3B there's this loop inserted here, whereas in the 3A it's a much shorter loop, and steric clashes would make it impossible for this domain to live next to that loop, and so it seems to have moved across. Okay, so that's the structure of a monomer, but if we try to understand this a little more, it helps if we can divide it into different domains. So we have domain 1, which forms the point of that assembled tetramer. Domain 2, which forms the body and makes the bulk of the interactions which hold together the tetramer. And domains 1 and 2 clearly are highly alpha helical. Domains 3, 4, and 5 are beta sheet-like, aren't involved in stabilizing the tetrameric interactions, and form those wings or propeller blades of the tetramer. And so we can clearly see a domain structure to this protein. But at this point, this is where the structure has really given us insight, which has helped us to understand how we think the protein is going to function. And what I'll do is I'll talk you through the different domains, but also remind you of that structure I showed you in the first place, of one of the best studied insecticidal protein families, the cry family, the 3-domain proteins. And what I'll try and show you is how we have, although there's no primary sequence similarity, there's a great deal of structural similarity between the two classes of proteins. So if we start with the domain 2 of the VIP3 protein, it's an alpha helical bundle with a hydrophobic central helix with some amphipathic helices surrounding it. And that is very closely mirrored by the domain 1 structure in the cry proteins. And if we look at this in terms of domains 1 and 2 from the VIP3 and the Cry5, what we see is that, again, you see the alignment is pretty good for the bundle of helices, but clearly we have this extra peak formed by domain 1, but there are also extra helices present in, or an extra helix present in domain 1 of the Cry proteins, and we can speculate that this extended form in the VIP proteins is perhaps a little like a slightly unfolded form of the Cry family of proteins. When we look at the domain 3 of the VIP, what we see is that it's a beta prism structure. So there's 3 sets of anti-parallel beta sheets forming a prism structure. And that is very similar to domain 2 of the Cry family of proteins. And this domain is known to be involved in the Cry proteins in receptor recognition. And there's some variable length and sequence surface loops down here at the tips of some of these beta turns, which are known to be very important in receptor binding specificity. If we look at domain 4 of the VIP3s, although there's no sequence homology, there is high structural similarity with its own domain 5, and also with the domain 3 of the Cry family. And this is a jelly roll twisted beta sheet structure that we see here. And again, in the Crys, it's been shown to be important in specificity. And it's also got lots of features of sugar binding groups. So both domains 4 and 5 have features which look as if they might bind to sugars. And also domain 2 has similarities with sugar binding groups and lectins. So that led us to speculate that we might be getting interactions with components such as glycolipids in the membrane. And we've recently been doing some work to try to probe that. And these are upper and lower phase charged and uncharged lipids from 2 species of insects, Spodoptera exigua and Spodoptera littoralis, which are targets for our toxin. And what we're seeing here is that we're getting binding of the toxin to those lipids. So there may be an interaction potentially via those beta sheet domains with the lipids in the membrane, which would potentially make sense in terms of facilitating interactions and pore formation. And we're currently applying for funding from Instruct to do some more cryo work with various ligands such as these to try and understand that interaction in detail. So what we have is, from simply seeing the structure and by comparison with other similar structural groups, the Cry family, we're able to come up with a model of how the domains may function. And that has domains 1 and 2 involved in pore formation, as we know they are in the Cry families, and domains 3, 4, and 5 involved in receptor binding and this specificity. And if we look at the sequence of the proteins across the VIP3 family, what we can do is look at the variability. And what this shows is anywhere where there's a bar shows that there's a potential variability in the amino acid, and the height of the bar shows us how many residues can be present at that particular position. And hopefully what you can see is that there's more conservation in domains 1 and 2. The bits which are actually functionally involved in setting up that tetrameric structure and penetrating the membrane, and less conservation in these domains 3, 4, and 5, which we think are involved in receptor specificity, and the toxin specificity. And this correlates with the fact that we have variability here, and between different members of the family, there are different target specificities. While we're looking at this, I just want to point out for later a little region down here around amino acid 20, which is highly conserved within this whole family. And I will come back to that in a minute. So what we have is a model where we have pore formation driven by domains 1 and 2, and receptor specificity driven by the other 3 domains. So is it a pore forming toxin? Well, when we add the protein to liposomes, we see very poor permeabilization. When we use the form that I've shown you, the structure I've shown you so far is that of the pro form of the toxin. And you'll remember that I told you that it gets activated with trypsin. And when we incubate with the trypsin activated form, we then get a nice permeabilization of liposomes in a dose responsive way. And we were able, in our EM work with Becky and the team in Leeds, not only to get the structure of the active of the proform, which I've shown you already, but also of this activated form of the toxin. And hopefully what you can see is that there's been a major structural rearrangement now. If I just remove one of the subunits here so that you can see the inside of the activated form a little better. What's happened is trypsin is cleaved in this exposed loop here, and that's freed this tip of the helix, tip of the tetramer, and part of the domain 2 shown in yellow here to swing around and come down and form a spike at the bottom of the protein. So really a big change in that whole system, and we've lost the peak here from the pyramidal tetramer. So if I show you that in a different context, here's this is not a model of the actual process. This is a mock-up in my hands of what seems to be happening on activation. This section seems to swing down between the blades of the propeller on this molecule and then open up a little bit like a collapsible walking stick into a long extended alpha helical spike coming down from the molecule. And when we look at our activated structure, we can only see part of that. And it seems that the ends of these extended helices are disordered in solution. So we're not seeing them clearly, but we are seeing the upper part of this. And again, from all 4 of the monomers within the tetramer, and they're forming this extended coiled coil. And if we look at that again, just in terms of the monomer, you can see the movement of this region down to the spike. But domains 3, 4, and 5, and the rest of domain 2 remain in essentially the same position. So the core is remained. That explains why the tetrameric form is maintained, because all of this, which is making the tetrameric contacts, is maintained, and these potential specificity regions aren't changing at all. If we look at the molecule from the top, in the closed form, in the pro form, the alpha helical domain 1 comes and sits with its partners at the top and forms a closed peak. But after that activation and the formation of the spike at the bottom, you open up a pore of about 5 angstroms, which is consistent with this protein being able to act as a pore forming toxin. And we went on to look at this protein actually on liposomes in those permeabilization assays. And hopefully what you can see is that as we scan up and down with some cryo-EM tomography, is you can see little palm trees appearing and disappearing here. And if you integrate those so that we can actually see them, what we see now is what we believe to be the pore formed in the membrane. And it's really interesting because what we have is this long shaft here with the bulk of the protein well proud of the membrane and just the tip apparently inserted. And if you remember, the structure of the activated form that we have is quite short in terms of this spike. It projects from the bulk of the molecule, probably less than half of the width of the molecule itself. Whereas here, we're seeing a very long extended shaft, more like my mock-up here with a long alpha helix coming down. And we think that what's happening there is that the interaction with the membrane is now stabilizing this coiled coil alpha helix all the way down to its base. And if we look at the what would be forming the pore here, it has to be, it's not a turn, it's not a loop, it would have to be the tip of the alpha helix. And if we look at the N-terminus of this protein, what we see is that region I pointed out to you earlier, which has got the high degree of conservation around amino acid 20, and it predicts to be a membrane spanning region. And furthermore, when we activate this protein, not only do we clip so that we can release the spike, we also cleave off the N-terminus of the protein so that this hydrophobic region becomes the very N-terminal end of the protein. So what we think we've got is a model where domains 3 to 5 are involved in receptor binding and potentially in interaction with sugar groups. We've got a proteolytic activation, which triggers major structural change. We've got the production of this spike with an internal pore to allow pore formation, mediated by just the insertion of the tip to cause cell lysis. And these are all factors that we want to explore more by further access to the cryo-EM facilities and production of mutants and looking at interaction partners. So this tells us now from a very basic start, a huge amount about how this particular family of proteins might function, all thanks to the cryo-EM. And it also has implications for how the 3-domain family of toxins work, which, although it's well characterized structurally, we have no knowledge of its pore formation. So we can draw parallels there. And finally, of course, to thank the people involved in this, all the guys up in Leeds, particularly Becky, who drove the structural work, myself and the people in Cardiff, and Mark Zach from Corteva, who gave us a vast quantity of purified protein to allow us to carry out the structural studies. So thank you very much. I'm happy to take any questions.
Hi, Colin. Thanks for that. That was really, really interesting. Great talk and some cool structures in there. While we wait for the question and answer session to see if anything comes through, I had a question. First of all, I know you said you were interested in lipids and you were looking to do some more work on the lipid things. One of the things I noticed is that, well, firstly, my question is, do you envisage a role for lipids to just be in targeting, or do you think there could be a role in priming the protein at some stage as well? And then the follow-up question is, in your structures, those domains that are proposed to recognize lipids are quite far away from the membrane. So how do you reconcile those things?
The answer to the second and the first question may well be related. So we think that there is a an interaction via those domains with the lipid. I mean, that remains to be established, but that's our hypothesis. Now, again, by parallel with the 3-domain family of proteins, what there seems to be is a reversible, relatively low affinity step, which concentrates the toxin up onto the target membrane, which is then followed by an insertional step, which becomes irreversible. And there's clearly a role for protein receptors in the specificity of VIP toxins, but a general interaction with the membrane to bring it to the cell surface may well be a feature. And one thing that we are trying to look at is whether the lipids that it binds are insect-specific lipids, or whether they're general lipids, or in which case there might be an element of specificity coming along, but the major specificity is going to be protein-based. We're also interested, obviously, in looking at whether glycoproteins are involved. Glycoproteins clearly could be further proud of the membrane as membrane active proteins, and so might be at a similar height to those domains. We're looking in an artificial system here of liposomes where there is no receptor, and by mediating the protein concentration, we can get pore formation. I've sort of lost track of whether I've answered all the parts of your question.
That's
Sorry.
Great. Thank you. I think Jamie has a question as well.
Sure.
Yeah, I really enjoyed your talk. I just wondered if you applied symmetry when you did your refinements, or you worked in C1, or you had a go with symmetry expansion.
As I say, I'm not the person who did the structural work on this. Becky and her team really drove all of this, but I believe that they did. Certainly, I mean, it's got C2 symmetry because it's a dimer of dimers, but I believe that they went through other variants and other options. And I think they also, I believe they probably did do some of the masking you were talking about, but I'm afraid I'm not able to comment on the niceties of that.
Yeah.
Oh yeah, I'm sure they did. I'll ask Becky next time.
Yeah. She will tell you in great detail.
We can go back and check, but yeah, it's an interesting one because it is C2 and then the bottom is C4, so there's a potential to explore a lot of different things with the symmetry expansion. So especially when it comes to lipids, it would probably be a really interesting thing to consider. Yeah,
and the activated form is properly C4.
Yep.
So you get the switch because of the loss of that peak and going into a properly tetrameric form.
Unless anyone's got any last minute questions that they want to fire in the Q&A, I think that's everything, and I think we should thank Jamie and Colin again. It's been a great seminar, so thank you guys.